Hello everyone, this is Mr. Schultz, and this is AP Biology video 3.1. This video covers cellular metabolism and is going to also cover Biology in Focus Chapter 6, and I apologize, this video might end up being a bit longer because it's covering an entire chapter instead of just part of a chapter. So let's start and take a look and talking about how our cells transform matter and energy. So metabolism is the totality of an organism's chemical reactions. So when we talk about metabolism, we're talking about all the chemical reactions within the body. Metabolic pathways are a specific molecule is altered in a series of defined steps resulting in a product. So metabolic pathways are going to be pathways that produce some sort of product. It could be building something or tearing something apart. Catabolic pathways release energy by breaking down complex molecules. So if you see a catabolic reaction, it means we're taking something big and complex and breaking it down to harness its energy usually. In an anabolic pathway, we're going to consume energy to build complicated molecules. So this might be something like protein synthesis is an anabolic pathway because we are going to take something small and make it big and complex. And energy released by one can be stored to power the other. So when we talk about photosynthesis and respiration, photosynthesis is an anabolic pathway. It creates something. Whereas catabolic pathway would be like respiration, where we take sugars and we break them down into energy, ATP, and then some waste products. So before we even get started on the actual metabolism stuff, let's talk a little bit about energy. Energy is the capacity to cause change in science. So there are a couple of different types of energy we look at in biology. There's kinetic energy, which is energy related to motion. Thermal energy, which is kinetic energy related to random movement of atoms, where we come up with the word heat, the transfer of energy from one thing to another. So thermal energy is just kinetic energy on an atomic level. Potential energy is energy that matter possesses because of its location or structure. So an example might be like a car at the top of a hill might have a lot of potential energy. Now it's not moving, but if you let the brake go, like the parking brake go, and the car rolls down the hill, it changes that potential energy into kinetic energy. And then finally, we also are going to be looking at chemical energy. This is potential energy available for release in a chemical reaction. So a lot of biology and energy is stored in these different molecules that can later be broken down into energy, which is going to end up being ATP. So couple of things. Laws of energy transformation. So we have what's called thermodynamics, which is the study of energy transformations when we change energy from one type to another. The first law of thermodynamics simply states that energy can be transferred or transformed, but it cannot be created or destroyed. So when we look at energy, it can't be just made out of nothing. It has to come from somewhere else. And so we have to keep that in mind. When we look at like where does most of the energy on the planet come from, well, it comes from the sun. Specifically, the sun is fusing small elements into bigger elements, and by doing so is releasing a whole bunch of energy, in this case both light and heat. The second law of thermodynamics says that every energy transfer or transformation increases the entropy of the universe. And entropy is a measure of disorder or randomness. So in biology, most entropy is going to be released as heat energy. And there's this idea that most reactions that are releasing heat energy don't harness that heat anymore, and it just gets released back into the universe. And slowly but surely, the universe is dying because most energy is turning into heat energy and will end up being spread throughout the universe. Now, there's some debate on this theory anyway, but it's just kind of an interesting thing. For a process to occur spontaneously, though, it must increase the entropy of the universe. So things have to go from more organized to less organized in order for a reaction to happen. And we like to think of that as like we have to add heat or energy into the reaction to get it to go, and it's generally going to do something where it's going to release organization as it goes through it. And it's a little complicated and a little weird, but you just need to understand that generally we're going to see an increase in entropy as we do reactions, and you can think of entropy in biology as being mostly heat energy. Now, we have what's called Gibbs free energy change. It's a mathematical equation that you might encounter on the AP test, but it's pretty rare to see it. Free energy is the portion of a system's energy that can perform work when temperature and pressure are uniform throughout the system. And only reactions with a negative G can occur with no input of energy. So these are what we call like our spontaneous reactions. So for a reaction to have a negative G, the system must lose energy during the change. Heat has to be released. And the equilibrium is delta G equals 0. So this is a state of maximum stability, and it's when the forward and backwards reactions are equal, and any change requires an input of energy. So spontaneous always moves towards equilibrium. So we're always trying to get towards a balance when it's a spontaneous reaction. But one of the things you have to keep in mind with equilibrium is that the reactions, when a reaction is just naturally occurring, is going back and forth between both products and reactants and doing the opposite reactions. And so a lot of reactions in our cells are controlled reactions because we don't want that equilibrium going back and forth. We want to actually take what's being made and use it in some way. So, free energy and metabolism. Exergonic reactions happen spontaneously. Their delta G is always negative, and they break bonds and give off energy. So when we see things breaking apart, they're generally exergonic reactions. Endergonic reactions need energy to take place. You have to put energy into them. And the delta G is positive, and so it creates bonds and absorbs energy. So like an example, endergonic reaction, it requires this much energy to push the reaction so that we can get our products. And for exergonic, what ends up happening is our delta G is negative, which means that our reactants naturally want to get down towards the products. And so that's where that delta G comes from. And if we look at the actual equation, delta G, the change in free energy, is the total energy in the system minus the absolute temperature times the change in entropy for the reaction itself. And to be honest, they really haven't asked a lot of Gibbs free energy questions on the AP test in a long time, and I'm starting to think that they're probably looking at possibly dropping it off of the AP bio test for the simple fact that it's just not used much on the test anymore. So, equilibrium and metabolism. A system in equilibrium has a delta G of 0. No work is being done, and the cell is dead. So if your cell has a delta G of 0, it means nothing's happening and your cell has died. A constant flow of materials into and out of the cell keeps the metabolic pathways from ever reaching an equilibrium. So no matter what, even if you would think that it would just go back to 0 at delta G and nothing would happen, the cell is constantly pumping more reactants into the cell and then constantly removing the products and waste out of the cell, so that the cell is always out of equilibrium and is always pushing towards that equilibrium by doing work. And catabolic pathways, they release energy over multiple steps. So instead of breaking down a sugar molecule all at once and having all this energy released, which would then get mostly lost as heat, we instead harness it by controlling each reaction as time goes on. So a product from one step is generally going to be the reactant for the next step in the chain. So an example would be like cellular respiration. What goes into respiration is glucose. By glycolysis, you end up with what's called pyruvic acid, and that pyruvic acid gets modified, and then it gets sent into the citric acid or Krebs cycle, where it then goes through a process, and then it comes out with products that are then moved to either the next part or released as waste. So products from one reaction end up being the reactant for the next. One of the big energy carriers in cells is what's called ATP, or adenosine triphosphate. An adenosine triphosphate contains the sugar ribose, with an N-base adenine, and a chain of 3 inorganic phosphates. So when you see that PI, what they're talking about is inorganic phosphates attached to it. It also serves as the nucleoside triphosphate used to make RNA. So when our cell needs to make an RNA chain, they actually use ATP, and then what they'll do is they'll change the nitrogen base to whichever one it needs. The bonds between the inorganic phosphates can be broken down via what's called hydrolysis, where we add a water and it breaks things apart, and it ends up forming adenosine diphosphate, ADP, and an inorganic phosphate that just kind of floats around till it needs to get put back on the ATP. The 3 phosphates are negatively charged and they naturally repel each other. And the release of energy during the hydrolysis of ATP comes from the chemical change to a lower free energy, not the bonds themselves. So like in freshman bio, we usually compare it to like the bonds are holding the energy. When we break them, we release that energy, but that's not actually how it works. We explain it that way because it's easier for freshmen to understand that like this bond is holding all this energy and not the molecule itself. Because when the molecule actually breaks, that it releases energy. It's not the bond that releases energy, it's the molecule itself. So how do cells harness energy from ATP? The ATP hydrolyzed by itself creates heat, which is super inefficient. An example of this is our body. When we get too cold, our body naturally starts to just break ATP apart to cause like our muscles to shiver, which increases the heat output of our cells, which then helps warm us up. So enzymes use the energy to drive endergonic reactions. So reactions that need energy will use an ATP molecule to run it. So we call it phosphorylated intermediate. So it has the inorganic phosphate attached, and it's now less stable than it used to be, so it is more likely to react. Basically, we take the ATP, we attach it via the phosphate breaks off, and all that energy the phosphate has now can put it into the molecule it's attached to, and that molecule becomes unstable and does some work. So transport mechanical work is caused by phosphorylation of a specific protein. The ATP is attached, the inorganic phosphate is left behind, the protein changes shape, and then the inorganic phosphate is then released. So this is how ATP works. It sticks itself to something else via a phosphate group. The phosphate breaks off. The phosphate gives its energy to the protein does the work, and when the protein's done with its work, it changes back its shape and the phosphate is released. And that phosphate just goes back into floating around the cell until it can be reattached to the ATP. So to regenerate ATP, a living organism is constantly using ATP, just constantly, and it recycles it. So it doesn't have to keep making ATP molecules every time it needs energy. Instead, it just reuses the same ones over and over again. And there are millions of them in our cells because our cells are constantly doing work and they constantly need energy. So ATP formation is an endergonic reaction. It is not spontaneous, which means that if you put a whole bunch of ADP and phosphates into a beaker, they would not attach to themselves naturally because it takes too much energy to do so. They don't want to do that. And so what we use is we need catabolic pathways to make ATP, to build it. And so cellular respiration and the light-dependent reaction of photosynthesis in plants both form ATP via catabolic pathways, where they actually take and harness energy. Respiration harnesses energy from sugar molecules being broken down, and the light-dependent reaction takes energy from light, exciting the electrons in things like chlorophyll to actually harness and make ATP. So one of the other things that we look at when we do for this chapter, is enzymes. Enzymes are going to be a special kind of protein. Spontaneous reactions occur without energy input, but they can take a really long time. So anabolic pathways like breaking down sugar can happen, but they take forever. And so our cells instead use what are called enzymes, which are macromolecules that act as a catalyst. They basically just speed up the reaction without being consumed. And so that's a really cool thing about enzymes is they're proteins generally, with a few exceptions, that take a reactant and cause the chemical reaction to happen without needing nearly as much energy. So most of them are proteins, with the exception being what are called ribozymes, which are made out of ribosomes, which are RNA, which is pretty neat too. And then without enzymes, many of our metabolic reactions in our cells would just shut down. Photosynthesis can't happen in plants. Respiration can't happen in all eukaryotes. Any other things like harnessing ATP, they can't happen without enzymes because enzymes are used to help make ATP. It's a huge issue. So if you have like an enzymatic mutation where an enzyme doesn't work correctly anymore, you can't do that metabolic pathway anymore, which can cause major issues. So the thing about enzymes is they're going to reduce activation energy. So all reactions require bond breaking and forming. In hydrolysis of glucose, a bond is broken between glucose and fructose and water. So what's going to happen is glucose and fructose are connected in a sucrose molecule, and we're going to break it apart. And we need an enzyme to do that. We're going to need some help from water too, so because water is used up. So the new bonds are formed to make a glucose and a fructose. And these two smaller sugar molecules can then be harnessed later to make like ATP in respiration. The activation energy is the initial energy for starting a reaction. This energy is what it takes for bonds to begin breaking in the reactants. So activation energy is how much energy it takes to start breaking the bonds apart in a reactant. Once bonds are broken, they can rearrange, losing their free energy because now they have all this extra energy. And the formation of new bonds release more energy than was invested in breaking the old bonds. So if you look at, you're going to see these a lot, especially when we do like our POGL activity. Here's our reactants. We need to have an input of energy to break bonds apart, make them less stable so that they can eventually go to a more stable product. And so this is how reactions work. We put energy into them, or at least certain reactions, endergonic reactions, where we put energy in to get something more stable out. And why are enzymes important? Well, enzymes speed up the reaction by lowering activation energy without increasing the temperature. Because you can actually increase reaction rates by increasing temp. But eventually, increasing the temp only does so much, and it could kill you because it could start denaturing our proteins. We don't want that. So enzymes speed up the reaction by lowering the activation energy. It doesn't change the delta G of the reaction because you can't change an endergonic reaction to an exergonic reaction. All it does is it just kind of reduces the amount of energy needed to input it by aiding the reaction itself. So enzymes are very specific to the reaction that they catalyze. They determine which process will be going on in a cell at any given time. An enzyme only does one job. Enzymes can't do multiple things. They're evolved to specifically only do one job at a time. So how do enzymes work? Well, the substrate is the reactant that an enzyme works on. So you need to know this word. A substrate is the reactant. The enzyme substrate complex is going to be the enzyme and its substrate that it works on together. When the two are joined, the enzyme converts the substrate to a product. And then the last thing you need to know is that enzymes have what's called an active site. It's the area on an enzyme that the substrate fits into. When the substrate moves into the active site, the enzyme will subtly change shape to lock the substrate into place, and we call this an induced fit. And every once in a while, an enzyme that's evolved to specifically work with one reactant might also have something else that can sit into this active site, either blocking it, inhibiting it, or could be like something that's going to block it completely, and instead of just shutting the enzyme down for a while, could shut it down permanently. And that happens sometimes too. So there are a couple of effects on enzymes as well. These are environmental effects, and we're going to look at a couple of them in our enzyme lab. One, we can change the temperature. Above a certain temperature, proteins start to denature. So there are specific enzyme temperatures for different enzymes. Some work better at higher temps, some work better at lower temps. But once you get above a certain temperature, which is about 105 or so in humans, a lot of our proteins start to denature. And so our enzymes start to break apart and they can't fully reform properly. And so we end up losing the enzyme and all those metabolic processes go away. Below a certain temperature, which is usually right around freezing, water freezes and enzymes aren't going to do any work anyway. There's not enough energy around to put into the system to have a reaction work. Now, another example of a environmental effect would be pH. Most pHs are the pH of most enzymes is probably between 6 and 8. So that's the optimal pH, right around neutral. However, there are enzymes that work in extremes. Pepsin is an example of an enzyme that's found in our stomachs. And it has to be able to withstand low pHs because our stomach acid has an incredibly low pH of about 2. So pepsin works really well around 2 to 3 pH, and that's it. And so different enzymes have evolved to handle different environments, but pepsin does not work well once you get it out of stomach acid, so it has to be in it. Cofactors are non-protein helpers. they're things that are going to help with the enzyme and get it going, or might be like a key, an activation key. So coenzymes are cofactors that are biological molecules. Sometimes like a specific biomolecule has to be present for the enzyme to actually work. Without it, the enzyme doesn't work at all. And then we have what are called enzyme inhibitors. There are competitive inhibitors. They block the substrates. And then non-competitive inhibitors, they bind to other parts of the enzyme. A competitive inhibitor is going to block an activation site by sitting in it. A non-competitive inhibitor is going to stick to some other part of the enzyme and change its shape so that the substrate can no longer stick into it and process it. Optimal conditions are those conditions under which an enzyme works best. And during our enzyme lab, which we're going to have with the yeast balls are going to break hydrogen peroxide down, and they have specific conditions they work best under. And we're going to try to explore some of those and see which factors work best. We're going to probably skip pH, though, because controlling it is really tough in the lab, especially for a high school level, even AP class. And pH tends to mess with the yeast quite a bit, and I don't want them to ruin the yeast balls because then they stop working correctly. So when we look at enzymes and regulation, there's what's called allosteric regulation. Allosteric regulation is any regulatory molecule that binds to an enzyme other than the activation site. So this is non-competitive inhibition. It can be an activator that turns on the enzyme, which would be non-inhibition, so it would be turning it on, or it could be an inhibitor that shuts off the activation site. So it could turn it on or it could turn it off. It just depends on the enzyme and the actual regulation molecule. Sometimes a substrate binding to one active site opens several more active sites on a multi-unit enzyme, and we call this cooperativity, where we have an enzyme where one substrate opens up all the other activation sites on the substrate. These usually require more than one protein complex in an enzyme. Finally, we have what's called feedback inhibition. When a metabolic pathway is shut down or shut off by the final product binding to an earlier enzyme in the pathway, we call it feedback inhibition. So the product from our reaction goes back and it blocks an enzyme earlier in the reaction to shut it down. An example of this is what's called the isoleucine pathway. A threonine is the initial product. It's a amino acid. And if the cell makes enough isoleucine, it starts to inhibit the initial enzyme in the pathway, preventing the cell from just keep on making isoleucine. We only need so much of it to make proteins, and so if we keep making more and more, we're using up all of our threonine, and we're making an amino acid we don't need anymore, and that would be bad. So instead, bad. Isoleucine instead just blocks the first enzyme and prevents the reaction from continuing until all the isoleucine's used up, and now we can go back to the reaction opening up. So where are enzymes found? Enzymes are not just randomly mixed throughout the cell. They have specific places they need to be. Enzymes are usually grouped together in multiple enzyme complexes. So the product of one enzyme is the reactant for the next. And so they're usually like in a membrane, and they're just like stacked, slowly but surely going down. And enzymes are often found within organelles for specific functions. So enzymes for respiration are found within the mitochondria. Specifically, there are enzymes within the matrix of the mitochondria for the citric acid cycle. And then there are a whole bunch of enzymes inside of these little folds, which are going to allow for ATP to be made. And so we'll talk more about that when we get to respiration. So this is the end of the enzymes video. I went kind of quickly. I'm sorry. I have limited time to make this one. And I'm sorry it's such a long video because it is one whole chapter. We'll talk more about this in class. If you have questions about feedback inhibition or activation energy or anything like that, feel free to ask in class, and we can definitely spend some time on it this next week.